So LC Columns, I like this talk. It does a deep dive in columns. It doesn't just give you information for the sake of information, but it explains how columns are made and why columns are different. And the most important takeaway from this is I will give you a list of my favorite columns. And these are columns that I know work really well. And in the column business, this is the hardest thing to deal with. Column companies make bad columns on purpose. They make half the columns in the catalog are terrible columns by today's standards. They have to make them for someone who validated a method back when that column was bought. So they can never stop making a column. They can never improve a column. Believe it or not, that's because of you guys. You guys are assuming the columns can be the exact same every single time. Let's say you're a pharmaceutical company, you generate a method on this particular column, you're running it forever, it's great, and the column company, the salesman comes to you today and says, oh, here's a new batch of columns you ordered, and by the way, you're gonna love this new batch. It's so much better than the last batch. We got rid of the old cast iron tanks, and you're gonna love this new column. Are you happy to hear that? If you're a pharmaceutical company, you're like, oh, no, you can't do that to me. I signed an agreement. If you have a validated method, it's like you signed your name to the FDA saying, I promise I will never change anything in this method. The column has changed. Your method is completely, you must revalidate. Validation, a lot of us not in the pharmaceutical industry are always making fun of pharmaceutical folks because they're always like, wah, we don't want to validate, we don't validate. It's like, shut up, just validate. You know what it takes to validate? Novartis publishes, it costs them $500,000 to validate a method and 6 months. 6 months and $500,000. So it is not a trivial thing to do, and they don't really want to redo methods all the time. So that is why they rely on the column company to be consistent. The exact same column that I bought 10 years ago. Now the challenge for the column company is they cannot improve that column. We think about continuous improvement. We all want to make our products better. No, you can't do that in the column industry. So here's the story I tell. Waters, 1978, came out with the Waters Microbonder Pack. It was fantastic. It was the best column, the best chemistry, it was phenomenal. 10 micron particles, irregular, plus or minus 5 micron was the particle size. It was incredible. So all these drug companies bought it and validated methods. By today's standards, that's a horrible column. So imagine you're making the Microbonder Pack. It's 1978. It's a great thing. A couple years later, DuPont invents spherical silica. We'll see this in a picture, but perfectly spherical. It's a no-brainer. This packs column much better than the irregular silica, so there's no reason to use the old stuff. So if you're making Microbonder Pack, can you change to spherical silica? No, that would change the column. You must now have another line of columns that Waters called NovaPack. So they're making NovaPack column spherical, Microbonder Pack irregular, making them forever, cranking through. 1995, the next big discovery comes in. It's called high purity silica. So high purity silica, it turns out silica has small heavy metal impurities in it, and they affect the chemistry, they affect the acidity, they affect peak shapes. So in 1995, DuPont said, we figured this out. We can now make pure silica. They take silicon dioxide, convert to silicon dichloride, distill it, and then convert it back to silicon dioxide. It's pure, no heavy metals, no brainer. Do you want dirty silica or clean silica? Of course you want clean silica. Can Waters change the NovaPack to clean silica? No, that would change the selectivity of the column. They must come up with another line of columns using this. So now if you're a column company, how many lines of columns do you want to carry? This is just a business decision because every column you have to have different lengths, different diameters. So if you have 20 different columns and each one in 5 different, you now have 100 different sizes that you need to stock. So you're very careful about making improvements. Let's say you make an improvement. It's like, hey boss, this is really great. Let's make the column better. Yeah, it makes it a little better, but we're not going to release it yet. Let's save up 5 little improvements before we go out there and release another column. So column companies make bad columns on purpose to suit our needs, your needs, customer needs. So once we realize that, all the columns are the same price. I'm going to teach you how to look at the catalog and find the good columns and not the lousy columns because you're paying the same price anyways. So that is a weird, it's not really a scientific story, but it's one of those, oh my gosh, when I learned that, it shocked me about the column industry. It's like, I can't believe you guys are making bad columns on purpose. And like, well, we don't like to call them bad columns. We like to call them historical columns, not bad columns. Okay, so now let's take a look at these silicas. This is spherical silicas up top, irregulars on the bottom. When LC first started, we used irregular silica. And this is it comes from a natural source. It's actually, there's an open pit mine in California, and they dig out these little animals that lived millions of years ago, these little silicates, and their skeletons are silicon dioxide. So they dig them up and they crush them and they sieve them, and all the good particles are used to make toothpaste. So when you brush your teeth in the morning, it's a 30-million-year-old animal that died so you could have clean teeth. Yeah, that's a pleasant thought, right? All this, yeah. Say it? Oh, yeah, you go with it, man. Tell me what's the...
Yeah, it's just like ancient cell.
Yeah, very good. So see, I'm not lying. You told me the truth. Okay, so all the good stuff was made to use, it was used to make toothpaste, and all the stuff that fell through the cracks, all the fines, were used to make HPLC columns. It's a true story, and this was wonderful material. Silica is silicon dioxide. It's a rock. It's sand, right? It withstands really high pressures. It has very high surface area. Most of the surface is on the inside. That's fantastic. So it's got all these great qualities. It was irregular. We didn't know any different back then. It was like, yeah, we'll pack a column, and that was great. That was a microbonded pack. 10 micron plus or minus 5 micron. So imagine that distribution, 5 to 15 micron. So then DuPont hops in and says, hey, we make stuff like silica. They figured out how to do this spherically, how to precipitate it or agglomerate it as a perfect sphere. So once the spherical silica came out, it was a no-brainer. You don't want irregular. Try pack this into a column. It doesn't pack very well. So here's my analogy. Let's say we're going to pack this room to the ceiling with stuff, but you got to use chairs and tables and desks and different sizes, different shapes. You'll probably stack the tables and maybe shove the chairs in the gaps, but let's face it, you're not going to do a great job packing this room full. We're going to leave a lot of empty space. Empty space is bad in HPLC. So then I say, let's do this again, but this time we're going to use basketballs. You're going to be like, oh wow, basketballs. They're all the same shape, all the same size. They pack together very uniformly. So we do a much better packing using basketballs. Then I say, let's do it again. This time let's use ping pong balls. Ooh, they're even smaller, more spherical, even smoother. They pack together more tightly. So this says we want to get away from irregular. We want to go to spherical for sure, no brainer. And once we're in spherical, the smaller the particle, the better. So that is sort of the mechanical part of it. Every column company makes an irregular column. Agilent makes one, Phenomics makes one. Everyone makes this piece of junk. And unfortunately, when you turn to the page in the catalog, like the Waters Microbonded Pack, it doesn't say, this is the worst column we've ever made. You might as well just pack mud into your column because it's as good as this. They say, oh, it's a beautiful column and it gives good peak shapes and you might consider a NovaPack, but it doesn't say it's a bad column. So you got to sort of read between the lines and realize, oh, that's a bad column. So the surface of silica is very polar. So silica is perfect except for one major flaw, and that is it's polar. We don't like polar surfaces because polar things would stick and never come off. So when HPLC first started, it was only normal phase. We had silica in the columns, and it was a very limited technique. If you injected an acid, base, or an alcohol, it would stick to that surface and it would never come off. We don't have the ability to pull it off. So column companies figured this out. They said, you know, if we recoat this column, if we change the surface from polar to non-polar, this would be a much more usable technique. So this is what they do. They start off with raw silica, called silica gel, and the structure is SiO, SiO, Si. Look familiar, right? This looks like GC stuff. All GC stationary phases, all GC septa, all HPLC columns, all HPLC column end capping. It's all silica chemistry. So we have this, this silica chain, and hanging off each silicon atom is a hydroxyl group. We call that a silanol. That silanol is very polar. It's also weakly acidic, which means the hydrogen can dissociate and leave behind a negative site. So column companies start with this and say, okay, that's polar. We don't like that, so let's make it nonpolar. So they start with a molecule. It's an octadecyl dimethyl chlorosilane. So silicon atom, 18 carbons. That's the octadecyl. Usually dimethyl, I'll use an R group for right now, chlorosilane. Chlorine is a good leaving group. So the chlorine hooks up with the hydrogen, says we're out of here, that's HCl. That leaves behind an SiO-Si bond. So this is covalently bound to the surface. It's not simply coated, it's bound. That's a stable bond. That's hydrolytically stable, thermally stable. So now we put a C18 here, we do the next same thing over here. Nice big C18, a couple of R groups, chlorine. But then we run into a problem called steric hindrance. A C18 is so big, it's hard to put another C18 in between them. So we leave behind some silanols. here's a shocking statement. If you just do C18 chemistry, just regular C18 chemistry, you will only cover half of the silanols. You leave half of them. So is that a problem? Well, if you have an acid or a base in your sample, that is a huge problem, because that OH, that's a bad guy. It's a hot spot, active site, silanol, we have different names for it. And the reason it's a bad guy is the hydrogen can dissociate and the site becomes negative. So imagine if you have an amine. Remember we said amine is the hardest thing to do by HPLC. Well, they're not that hard to do. The reason we say they're hard to do is an amine in solution, if it ionizes, if you have a low pH, it is positively charged. You have a positive charged analyte, you have a negative charge on the surface, and guess what happens? Coulombic attraction, they stick and they do not let go. Adsorption is not symmetrical. It sticks and it takes it a long time to let go. That's why I refer to this like Velcro. Easy to stick, hard to pull apart. So if you inject an amine, you're gonna get a 5-minute tail. Looks horrible. So column companies know this. They realize this is big business, right? I mean, think about everything you guys are running, besides with the polymers, are probably acids, bases, alcohols. So they figure out a way to cover up the residual silanols. There's a couple approaches. One approach, most popular approach, is called end capping. So end capping means we're going to do the C18 chemistry 2, 3, 4, 5 times. We're going to do it, put down as many C18s as we can, and then we're going to come back around with a small molecule, trimethylchlorosilane. So this is a silicon atom, 3 methyl groups, and a chlorine. The chlorine and the hydrogen leave, resulting in the SiO-Si bond. And since this molecule is small, it could reach down to the bottom and cap off all the hot spots, all the bad guys. So if you end cap this, if you remove all the silanols, your peak, your amine, is beautiful, perfectly symmetrical. There's no reason for it to stick. And the reason is, if I get rid of all the negative sites, even if your molecule is positive, there's nothing to stick to, right? You only have half the Velcro, it's not going to stick to anything. So end capping is a very popular approach for getting rid of these silanols. There's another approach out there, and I'll show this one because this was invented by Jack Kirkland, the guy who wrote the big book, and really a pretty important guy in the field. Both Kirkland and Snyder passed away in the last couple of years. Snyder 2 years ago. Both of them had like really cool memorials at PitCon. So Pittsburgh conference, we had like huge parties for them, which is sort of neat. Pictures of them all over the place, and just a bunch of geek chemists getting drunk and celebrating the masters of HPLC. So Jack Kirkland did not like end capping. Jack Kirkland invented the first C18. He invented spherical silica. He packed the first column, and he said, "I don't like end capping. It's a dumb idea." Keep in mind, most companies end cap, but he's like, "I don't like it because if you really study chemistry, the longer this chain, the more stable this bond. 18s are the most stable column, C18. C8, well, almost stable. C4, much less stable." He said, this is a C1 column, right? This is a C1. That thing is going to hydrolyze, and it's going to become polar again. So over time, end cap columns lose their end caps, and they get more polar. So he said, I have a better idea. Instead of a methyl group here, let's put a big bulky side chain, like a T-butyl group. So imagine if I put a big bulky side chain here and a big bulky side chain here. The silanol is still there, but if I could effectively cover it, shield it, then it's as if it's not there. So this led to a series of columns that DuPont made. DuPont had a group called Zorbax. Zorbax got spun off and got bought by Agilent. So Zorbax makes a series of columns called the SB columns, stable bond. So they call this stable bond technology. So they make what's called the SBC18. They also make SBC8, SBC4. So stable bond means they've covered the OH and not end capped it. Since then, they have, since end cap, Agilent does end cap some columns, and they have some that are end capped and sterically protected. So back in the day, I always wanted to ask Jack Kirkland, so were you wrong about end capping? I see you guys are end capping now, and I'm sure he would say, I was never wrong. Those marketing fools made me end cap. So here's my approach. I say choose a good column company and then ask them for their best column. I don't care if it's end capped or sterically protected. I look at it as, you guys are big boys. This is a big business. Give me your best column. So every column company makes a series of columns, at least one, well, they make several columns. This type of column is called a base deactivated column. It has been deactivated for bases, right? We've covered up all the silanols. So let me tell you the great news. A base deactivated column is the best column for bases. It is also the best column for acids for the same reason. And it turns out to be the best column for neutrals because neutrals do not care. If you're analyzing benzene, toluene, xylene, they have no ability to stick. They don't care if their silanols are present. They'll give you the same chromatography. So my feeling is we have one column that's the best acid, best for bases, best for neutrals. That is our primary column. That's our one-stop shop. So when you're doing method development, when you're choosing columns, start with the best base deactivated column you can get. And that's going to do everything all the other C18s can do. And on top of that, it's going to be able to handle amines and ionized bases, ionized acids. It's going to look great. So like I said, every company makes a series of these. I will give you a list of my favorite columns. And I do this at great risk to myself because I feel terrible doing this. There are columns that I love, and I'll tell you what they are. And I feel bad for all the columns that I have not tried that I would probably love. So I apologize for everyone out there that if I don't mention your column, I'm sorry. But well, that's the way it is. So let's start with waters. Waters, I will sometimes make fun of some of their equipment. Sorry, Alliance, I got to make fun of that. But they make incredible columns. They have incredible chemistry. So in the waters world, they have one called Sunfire. So Sunfire is a classic silica, well end capped, great peak shapes, great efficiency, beautiful column. So water Sunfire. They also have another column that I like, and it's called X-Bridge. X-Bridge. Keep in mind that when you buy a column, the way we typically name it, we name it for the silica, Sunfire, and then the chemistry. So you have a Sunfire C18, Sunfire C8, Sunfire C4, Sunfire Cyano. Same with X-Bridge. So this is the name of sort of the silica, and then we add the name of the chemistry on top of that. X-Bridge was a really fascinating idea. It is what we call a grafted polymer. It's not pure silica, it's silica plus carbon, and it gives it very high pH stability. So most HPLC columns, most of the columns are limited to pH 2 to 7. Don't go below 2, you chop off C18s. Go above 7, you dissolve the silica. So pH 2 to 7, I still recommend you stay in that range. I don't care what column you have. If you stay in that range, you will live a long, happy life. Right? So 2 to 7. Below 2, we chop off the C18s. We hydrolyze the C18s off the column and the end caps. Above 7, we dissolve the silica. Silica is very soluble above pH 7. So that just ruins the column. So we stay within that range. Having said that, X-Bridge is spec'd all the way up to pH 10. Wow, crazy. So they did something very special when they made it. I do not like their Xterra column. I'm sorry. I know it's really popular. I know they sold a lot of them. Water's Xterra column was a cool idea that missed the boat. Their idea was the grafted polymer, it has very high pH stability, but it has terrible efficiency. I'm not saying it, it's not me saying it. Look at Water's data. They'll tell you the efficiency of that column is half of what it should be. It got sort of screwed up. So Sunfire, excellent. X-Bridge, excellent. Okay, in the adjuvant world, I got to list their SB-C18 first, because the SB-C18 is their #1 selling material. This is the stable bond. This is the putting those T-butyl groups for the steric hindrance and shielding the sites. The cool thing about the SB is this is the only column spec'd down to pH 1. Every column in the world is spec'd to pH 2. This one's spec'd to pH 1 because they don't have the weak link. There's no end caps on it. So if you came to me and you said, oh man, I really got to run at pH 1, I'm like, oh, I got the column for you, SBC18. It's also the only column spec'd to 90 degrees Celsius. Every other HPLC column, we spec it to 60 degrees. We say don't go above 60. For the SB, it's spec'd to 90 degrees. Why? Well, again, the weak link is missing. So it's neat. It's a great column. So if you're using this, there's no reason to change. I'll tell you my favorite column is called the Eclipse Plus. C-L-I-P-S-E, Eclipse Plus. So Eclipse Plus, I started using it maybe 7 years ago, and it's my favorite column that I've ever used. It is classical spherical silica. It is nicely base deactivated. And by the way, all these are the same price, so I'm not telling you to buy a more expensive column. Even the horrible columns are the same price as the best columns. So this is Eclipse Plus. I think it has a longer name. It's part of the XDB. It was the XDB Eclipse Plus, but I think they finally extra densely bonded with XDB. So they had a series called XDB. If you were using one of those, it predated this. It's fine. There's no reason to change. Those are all good enough. But this one's my favorite. Okay, in the Phenomics world, their number one selling silica is Luna. Luna is older, but it is incredibly popular. It is, like I said, their number one selling silica, and it is an excellent material. Spherical, classical, end-capped, good, lasts a long time, good peak shapes for bases. It's a good one. There's a new version of it. Well, my friends at Phenomics say, hey, if you like Luna, please switch to our new one, which is called Gemini. I will admit that I haven't tried a Gemini column yet, but I trust the people who I talk to there, who I've worked with, and they're like, hey, this is our new column. So let me tell you a cool little story about Phenomics. A lot of people aren't old enough to remember their beginning. So I'll tell the story. Now, I'm not dissing them. Not dissing them. I like them. I used to write classes. I wrote all their GC classes. But their beginning was a little sketchy. So the owner of their company, I've met him, very smart guy, Fasha Major. He is an architect by training, a brilliant architect, a brilliant marketing guy. He has no chemistry background at all. He saw this LC column business, and he said, someone told him, it's like, yeah, we spend $10 making a column, we sell it for $500. He said, I need to be in that business. So I'll just hire a couple of geeks and get them to reverse engineer the top column in the world. So number one selling column in the world was Micro Bond Pack by Waters. He's like, no problem. Reverse engineer the column. So they reverse engineer it, and he sold it, and he called here, instead of Micro Bond Pack, he called his Micro Bond Clone. So he didn't hide it. He was like, oh, I admit, I just copied their column. And my column is not better than theirs. It's just as good as theirs. It's not any better, but I'll sell it for half price. Like, whoa, what? Oh, and I'll guarantee it. If it doesn't work, send it back. I'll give you money back. People are like, whoa, I'll take a flyer on that. And people start buying it, and it's a beautiful column. It worked just as well as the Micro Bond Pack. Waters was furious. How dare you take our name in it? We're going to sue you. They take him to court. The case gets dropped right before it goes to trial because, I don't know, my professor was involved with some of the data, and maybe some of the data was about to come out that the Micro Bond Clone was a little better than Bond Pack, and Waters decided, you know, these guys will probably go out of business. Let's just let it lie. So they walk away. So now Phenomics making a boatload of money. So what's the next number one column in the world? Hypersil. So they make the Hyperclone. And they get sued again, and they win the lawsuit, and they keep going, And finally, they came up with a column called Prodigy. And when Prodigy came out, it was arguably the best LC column at the time. And everyone's like, whoa, this is amazing. Whose is this? Who do they copy? Who do they steal? Hey, Waters, is it yours? No, it's not yours. Was it yours? No, it's not. Well, they made their own column. They got so good at reverse engineering other people's columns, they're like, hey, we could sort of figure this stuff out. We can make some improvements. So I believe Phenomics is responsible for raising the bar in the LC industry, because we were sort of dead in the water. We were like nothing new for 10 years when Phenomics came out and started doing this stuff. So here's the funny part of the story. So Waters makes an Xterra column. Grafted polymer, super high pH stability, cool idea, but it had terrible efficiency. So Phenomics went to copy it, and as they're copying it, they're like, they made a mistake. They forgot. They fixed it, and they called it Gemini. So Waters finally got so mad, they're like, okay, we're not going to sue you. You know what we're going to do? We're going to copy your column. They copied it, and that's XBridge is a copy. And now there's a Gemini 2, which is a copy of a copy of a copy. So I think it's hilarious that these guys are button heads all these years, and they're finally like, hey, you know what? We're going to copy you this time. So Agilent, the third biggest player in this market, stood back and said, let's just watch my two biggest competitors throw mud at each other, and let's just see how this works out. So they sort of took a very neutral stance to this. I will say that customers love Phenomics, and they should, because they're very customer focused. The other people in the industry hate Phenomics because you guys like them, or I don't know. So they're sort of not hated, but sort of hated by the other big players. Phenomics is probably the biggest HPLC column company in the world. They are privately held. They don't have to report numbers, but we all know that they sell more LC columns than anyone else. So always worth paying attention to. I say they're customer focused because I was teaching an in-house course for Pfizer at a facility. This was Ann Arbor, Michigan. I don't think it's still there, but it used to be. They had a R&D facility. And I'm there, and I'm doing HPLC method development, and I ask, what kind of columns, what brand of columns you guys use, just so I'll be consistent with my slides. And they said, well, you know, we got a Phenomics cabinet, so let's pretty much use their columns. And I said, what's a phonomics cabinet? She said, oh, well, when we were building our new facility, phonomics came to us and said, do you mind if we put a big metal cabinet somewhere? You just choose a place. We'll put it in there, and we're going to stock it full of all of our stuff, all of our columns and SP. We're not going to charge you for any of that. Put a couple $100,000 worth of stuff in here, and anytime you want a column, you go to the cabinet, you take the column. You use it in the lab. If you like it, well, we're going to charge you for it. Eventually, we'll do inventory once a month. If you don't like it, no problem. Just put it back on this bottom shelf. We won't charge you for it. So I think it's brilliant. It's what I call the heroin dealer approach to marketing. It's like, hey, little kids, try a little bit of this and for free. And once you like, you know where to come back. Because they realize if you're a pharmaceutical company and you write a method on that column, they're going to sell that column to every one of your labs around the world for the next 20 years. So I think it's really smart. They will bend over backwards giving you free stuff and helping you out. And that to me is how a company should be. So yeah, so okay, sort of a cool story about phonomics. Yes. Okay, yeah.
So one question I was thinking of, so with the end capping, is that almost like a planned obsolescence then? If they know that over time, like this is going to get worse, well, then you need to come back.
Yeah, no, that's a good question. No, a good question. So first statement, I think end capping is fine. They work, they last a long time. Eventually your column's going to wear out for some reason. Maybe it's because you've lost some end caps, maybe because you've dissolved some silica or your column got crapped up. So I don't think that's the reason. I think so. Jack Kirkland was a purist, and he's, like I said, he's in the trenches every day, and he's like, why put on something that you know is going to fall off? Why not just take a different approach? So no other companies, I don't say no other companies, but he was sort of alone doing that. He sold a lot of them, and they still sell. It's a great column, but not a lot of other people sort of jumped on that steric hindrance bandwagon.
And there's no intuitive real difference in shelf.
No. Columns last forever. Yeah. A shelf lasts forever. There's hydrolysis, but no. I mean, not at normal pHs. Okay. Yeah. They'll last forever on the shelf.
Thanks.
Good. So, like I said, I apologize for the other folks. You know what? Who I should put up here is Thermo, Hyper Gold, Hyper Elite, not Hypersil. We have some Hypersil columns. They're old-fashioned end capping. Hyper Elite and Hyper Gold are their high purity silica columns. Okay, questions about columns? Did I leave anyone have a great column that they want to mention they want to put up here? I should do that. That's what we have, like a community sourcing where we're coming up with the name of the community. We're going to have everyone join, like you could ask questions and we'll all be together. So the current name, Axion Avengers? Oh, we don't know. We haven't decided on a name yet. Oh, okay. Toss it out. Axion Prime. Oh, okay. What do you guys think? Axion Prime? Yeah. So we should do something with all the folks on the internet, and we'll tell them to vote, and they'll get a free cup of coffee or something like that. Awesome. Yeah.
Prime is a completely open word right now.
Yeah.
Okay. Yeah. Okay. Axion Prime. Okay. I like that. So this just is the slide version of my drawing the chemistry. Just a little neater version of that. So all the columns are made the same way, whether you have a C18, C8, cyano, amino, diol. It's something hooked up to a silanol, to a chlorosilane, and then hooked up onto the column. This is just to remind us what end capping looks like versus steric protection versus no end capping. So we really want to cover up that hydroxyl group. Even if it doesn't make our peak shapes look bad, it gives us what we call a mixed mode mechanism. So there's more than one thing. Instead of just interacting with C18s, we also have a polar interaction, and it's best to keep things simple. So find a column with no silanols, and that is your best reverse phase column. So these are the different columns that we make. Like I said, we make all of these the same way. C18 and C8s are the most popular. The reason for that, I look at it as the mechanism is called hydrophobic interaction, and we seem to like this mechanism. It seems to work for 85% of the people in the world. So since we like the mechanism, shouldn't we find the most hydrophobic column? And that's what the C18 is. It has the most hydrophobic character. It does the most of that mechanism. Honestly, the C8 is almost the same. I know people are thinking C8 and C18, that sounds dramatically different, but here's how I explain it. On a polarity scale, where if water's here, Where would you put octane, C8 on the scale? The opposite end. Where would you put octadecane? Right next to it. So here's water, here's C8, here's C18. The separations are almost identical except a little less of it. So if this is a C18, this is a C8. C18, C8. So it's a little less interaction, a little less stuff. I would put these as almost interchangeable. In fact, I just say just buy C18. C8s are not as popular as 18s, but if you end up with an 8, it's the same. Same thing. C4 is very different. It's much, much less retention. Why would we ever want less retention? Well, I've got an idea. If your molecule is too big, too nonpolar, too many carbons on it. So that is, a good example of that is proteins. Proteins are huge molecules. They stick to the C18. They don't want to come off. We'd have to go to 100% acetonitrile, which would denature the protein. So we go to a C4, very short chain, and the proteins don't hold on as long. I've used a C4 chain for other things. I think I mentioned for silanosol. If someone gives me a really, really nonpolar compound and it takes an hour to come off a C18, well, it's going to come off a C4 a lot faster. So definitely not the first choice, well, unless you're doing proteins. Proteins, here's your first choice. C4, what we call wide pore material. Wide pore, I'll explain that in a bit, but that's your protein column if you're doing reverse phase of proteins. Otherwise, we go C18. Cyano and amino, these columns are different. I'll leave it at that. We'll talk about those during method development. There's not as many applications out there. Cyano was the, if C18 doesn't work, try cyano. It was just a, if this doesn't work, try something on the opposite end of the spectrum. Amino columns are now back in popularity a little bit for HPLC. I'll explain HPLC in a bit, but they are the go-to HPLC columns, sugar separations on amino columns. They can be used in normal phase or reverse phase, different mechanisms, but it's the same column. And then the ion exchange and the size exclusion, these are all non-silica based particles. These are based on a polystyrene divinyl benzene, a plastic particle. So this explains the mechanism. Mechanism is called hydrophobic interaction. And the way I think of it is we have the C18 is very nonpolar. And it will interact with the nonpolar part of your molecule, with the carbon in your molecule. So the more carbon in your molecule, the more interaction, the more it likes the C18. So I sort of say it dissolves into the C18. It doesn't really dissolve, it partitions into it. But the more carbon you have, the more time it's going to spend with the C18. So mechanism is called partitioning between the methanol and the C18. Water plays no role in this mechanism. Water only serves to dilute, to weaken the methanol. The reason I say that is because if a lot of people think this is differential solubility, in other words, what if my sample is really, really soluble in water? Shouldn't that make water a strong solvent? And the answer is no. Water is never the strong solvent, and it's a little hard to explain this one, but we could prove this. And that is with something like acetic acid. Acetic acid loves water, right? It would much rather be in water than methanol. But when we do acetic acid on reverse phase, when we add more water, it takes longer to come off. So if this were a solubility thing, it would come off earlier with water. This is not solubility. This is interaction between the C18 and the methanol, and the water is just diluting the methanol. So that's not a big deal, but I say that to a lot of people will be like, no, this is the solubility things. And if you think the solubility is going to take you down the wrong path. So water is always weak. So the stationary phase is C18, octadicyl silyl is a C18, octyl is a C8, cyano and amino. Those are actually, the real name is cyanopropyl aminopropyl. So they do have a carbon chain. They've got 3 carbons. So they're doing some nonpolar interaction, and then they have a semipolar head group, right? They got a cyano or amino. So it is a different mode. They are sort of holding on to polar things a little longer. I'm not opposed to them, but I'll show you them in method development. My feeling is you start with a good base activated C18 column. That's going to solve most of the problems. And then I'll give you alternate columns to try next. They'll be on that list. Mobile phase is water plus some organic solvent. Water plus methanol or acetonitrile. Those are your choices. A lot of people ask, well, why not isopropanol? Yeah, there's a lot of other stuff. Ethanol, I actually did a paper on using ethanol as a mobile phase. It's really cool. You could drink the effluent. I use a martini glass as my waste bottle, and I use a vodka bottle as my mobile phase. I have 2 kids, and we publish it together. I'm like, am I a good dad or a bad dad by letting my teenagers publish with a vodka bottle? So, but the bottom line is ethanol is not quite as good as methanol. So methanol and acetonitrile both work great. Isopropanol, it's too viscous. It works, you can use it if you want, but your pressures are going to be quadrupled with it, so why bother with that? Buffers and modifiers we'll talk about in a little bit. Okay, so C18 on acetonitrile or methanol seems to be the right selectivity for most applications in the world, for 85% of the applications. So reverse phase is the first choice for neutrals, polars, and nonpolars with molecular weights less than 2,000. It's also the best choice for homologs and benzalogs. You know what homologous series is? A series of molecules that differ by the number of carbon atoms. Methanol, ethanol, propanol, butanol. That is a beautiful example of what would separate by reverse phase, because every time you add a carbon atom, you add a lot more hydrophobicity. So it is guaranteed separation, homologous series guaranteed to separate by reverse phase. If you make any changes to the molecules, if two molecules are identical except this has an extra carbon, easy separation. This has an extra hydroxyl, easy separation. This has a double bond, this one doesn't, easy separation. So anything you change here is going to change its hydrophobicity. Weak acids, weak bases, strong bases, proteins and peptides. Things that are harder to do, yeah, we put amines on that slide. I sort of laugh when we do that because, yes, in 1985, amines were a big pain in the butt. But now we have great ways, great approaches for amines. Good base activated column, you're good to go. This talks about pore size. So pore size we have not talked about yet. We've been talking about particle size. 10 micron, 5 micron, 3.5, 1.8. But now let's talk about pore size, and I'll start off by saying the only people who need to care about this is people who are working with really big molecules, greater than 4,000. In my mind, this is for proteins. So I don't know, in my world, everything is less than 500 or less than 1,000. When we got to 4,000, I'm like, oh, you must have a peptide or a protein or some kind of a biological molecule. So this is for protein people. You need the pore of the packing material must be big enough to let the molecule inside. So here's the packing. Most of the surface is on the inside. This is like a sponge. 95% of the surface area is inside the particle. So your molecule needs to fit inside the pore to see all the surface. If your molecule can't fit in the pore, it gets excluded. We get really broad peaks and lousy separation. So in this case, we have got a bunch of probes. We have a normal size. So regular silica has an angstrom size, and if you look at the specs in your column, it'll say what it is. I always joke, no one knows what their pore size is. It ranges from 70 to 120. Those are sort of extremes. So 80, 90 is a typical pore size. So this material is 80 micron. So everyone makes a normal pore size, 70 to 120, and everybody makes a 300 pore size. So it's wide pore, but everyone's wide pore is 300 angstrom. So these probes, 500 molecular weight, it doesn't matter. 1,000 molecular weight, doesn't matter. 3,500 doesn't matter. 12,000, whoa, we see a big difference. And now we're seeing that exclusion happening. So these numbers, this is a guess. This is just a ballpark number. When we say greater than 4,000, some people say 2,000, some people say 4,000. The reason we don't know an exact number, it's not the molecular weight, it's the shape of the molecule. So if your molecule is 4,000 molecular weight, but a straight chain, like a snake, it could fit right into that hole. If you're a 4,000 molecular weight, but you have that tertiary structure, like a protein, and it's a big ball, it's not going to fit in the hole. So it's sort of a ballpark number. If you're less than 4,000, non-issue. Greater than 4,000, yeah, you should think about this. In general, we prefer smaller pore sizes because the column is more robust, a little higher efficiency. If we need large pores, we get large pores. This explains why we don't just choose large pores to begin with, because the columns are not quite as good. They don't last quite as long. They're more susceptible to crushing, but it's the way to go if you're using proteins. And here's an example of a large molecule with a normal pore and a wide pore material. We'll see we get much better peak shapes, skinnier peaks on the wide pore. Retention order in reverse phase is pretty straightforward. We know that the more carbon you have, the more interaction you're going to get. So the more carbon, the more retention. Less carbon, less retention. You start to branch the chain, we get a little less retention. You start to add double bonds. We're tiptoeing towards polarity, so we get a little less retention. We go to amines, aldehydes, ketones. So now we are in the polar world, so even less retention. The alcohol group, even more polar, and the most polar group is going to be the acid group. Those are the least retained. So we could look at a simple series of molecules and predict their elution order. So if we look at this first problem set with 4 compounds. They're all 4 benzene rings with something hanging off it. So we got benzene, we got benzoic acid with an acid group, we have toluene with a methyl group, we have phenol with a hydroxyl group. So the first question, which compound is going to come off first? Which one of those is the most polar? And that is number 2, the acid group. What's the next most polar? Number 4, very good, the phenol. Next one is a little confusing, so I always ask, what peak's going to come off last? Which one has the most carbon on it? Yeah, toluene is last, so the third one is benzene. A lot of people get confused, but you realize that if I add a methyl group, I'm actually making it more nonpolar. So the correct answer is 2, 4, 1, 3 in terms of their elution order. So over here, this is a homologous series. Actually, I'm going to give this problem set to Morgan because Morgan knows more about short-chain free fatty acids than anybody. So, all right, this is your life, isn't this? So, hold on. Acetic, propionic, butyric. Okay, if you want to smell some nasty smelling stuff, butyric acid is vomit. It's a vomit spill. Yeah, you want to grab some and pass it around? Yeah. And then it gets worse. Then we get the C5. Oh, what's C5? Valeric? Valeric. Yeah. So valeric smells like sweet vomit. So it's even worse. Is something worse than vomit? Yeah, there's something worse than sweet vomit. So these are all sort of nasty smelling. Okay, so their elution order, number 2 comes off first, number 3, number 4, and then number 1. Okay, can I tell you a quick funny story? Can I mention your project? Okay, so this is a story, short story, and I called the name of the story is A Shitty Day in the Life of an Analytical Chemist. I didn't say a bad day. I said a shitty day in the life of an analytical chemist. So I do some work for IIT, for Illinois Tech, new name. I'm an adjunct professor there. I teach this course for them. And I also teach some other stuff. They'll have me come in and give lectures. So there's a really sharp professor, Professor Amandeep, and she is a food science professor, and I go there once a year and I give a GC lecture in their food science classes. So she called me up and she said, hey Lee, we're in a bind. I have a graduate student, she's working on a project and the instrumentation is dead in the water. We have GCMS spec, it's not working, we've moved it and we don't really have plans right now to fix it. So I'm a little worried about this graduate student because she's doing short-chain free fatty acids by GCMS spec, and she wants to measure them and she can't do her dissertation, she can't graduate until she does a project. And I said, oh yeah, I'd love to help out. And I said, I have 15 GCs, FIDs, happy to loan you one. GCMS specs, I really have one, so I'm a little not so thrilled about loaning that one out. But you're using free chain fatty acids, yeah, that'll be fine on FID. She's like, oh my gosh, that's amazing. She's like, I can't thank you enough. This make a big difference. And she's like, we'll get together with the students. And she goes, so free chain fatty acids. I said, yeah, no problem. She goes, I'll buy the standards. She goes, we'll do the sample prep because you take the stool samples and then we grind that up. We extract them in a buffer. I'm like, what kind of samples? She has a bit of an accent. And she said, the stool samples. We'll take the stool samples and we'll extract them. I was like, are you suggesting you're going to inject poop on my GCs? And she was like, we're going to do the extractions here. I was like, let's get that clear. No extractions on my. She goes, no, we'll do all the extractions there. So, so it's to look at free chain fatty acids in fecal material, human fecal material. And I'm thinking, it's like, wow, okay, whatever. I've done all kinds of ways. This is sort of a wacky project. So I started doing a little research on it. She sent me a paper. I started researching it. And I read this paper from a medical school, and they're talking about free chain fatty acid profiles being indicative of colorectal cancer. That you could discover cancer by looking at this, and also other. There's a couple of other celiac disease and all the that are based on the free fatty. It's like, oh my gosh, as funny and weird as this project sounds, this is like it's going to cure cancer. So I sort of you poop in a box and it says you've got cancer. It's like what a great. So my brother is a sort of famous oncologist at University of Chicago. He's a professor of oncology of GI stuff. So we were hanging out on the weekend. I was like, hey, I got this project coming up, and he goes like, oh my gosh. He goes, that's hot stuff. That's like, yeah, that's for real. That's for real. I think I'm in, and the person's name is who's in charge of this project is someone you all know, and their name is Morgan Freeman, which is Morgan's name. So I thought that was sort of a funny part. It's like, and I'm looking at the emails like, did I just get an email from Morgan Freeman? And yeah. So how cool is that? Yeah, so that's your project, and we are actually doing that project here, and we've already stumbled on a really cool thing that improved our sensitivity like 30-fold. So we're already sort of making some neat improvements to that. Yeah, question.
So we're doing the caffeine lab this afternoon today. People brought in their own samples. Next boot camp, can we do short chain prefabricated?
Absolutely not. Yeah. So you want to know details? You want to know some more? No. Okay. So it's funny is that it reminded me of a story years ago when I was in graduate school. I was a senior graduate student from McNair. We used to teach the ACS short courses 4 times a year, course sort of like this. We made very little money. Like we never had money as a group, but we loved each other. We had great, like we got donations of all the best equipment, but McNair never wanted to do the big, the big money projects. So I had an idea for a moneymaker. I was like, let's do one more short course, but we won't do it for the AD, we'll do it for ourselves, just pure profit. And we'll do method development and we'll tell people, bring your own sample. Imagine, industry, bring whatever sample you've been working on for years, you can't figure out, bring it to the course and we'll work on it. So awesome idea, right? And we get a bunch of people sign up, we're gonna make a boatload of money, so we go around the room. One guy works industrial hygiene, nuclear power plant, and he has to, they have to watch their employees, make sure they don't get too much radiation, and once a month they get stool samples and they analyze them for radioactivity. So he's talking about this, I'm like, am I to understand that you brought radioactive poop with you? And he's like, well, I left it in the car. I was like, let's leave that in the car for now, and then we'll talk about the approach. Yes, so that has always been in the back of my mind when we got this sample. So, cool. It's actually really neat. It's a very neat project. Okay, so thank you for letting me share that with them. Okay, so where are we? We're talking about columns. And this is a great slide. I've already told you this one, but this is a nice slide. And this is an easy way to cut your analysis time in half. We call this method optimization. You have a method that works. That's the important word, that works. So if your method works, here's how I look at it. You've got a working method. You must have found all the conditions to give you resolution. So remember that means it's a resolution equation. So you must have found the right capacity factor. You must have found the right selectivity, and you must have the right efficiency for your separation. So if you bring me your method, this is one of my favorite projects, we do this for companies, I will then return a method to you that's faster. So, and this is really easy. Don't tell my customers this is easy. You can charge a lot of money for this, but this is really easy. They did the hard part. Of these 3 terms, do you know how to adjust capacity factor? Sure you do, solvent strength. You guys know how to adjust efficiency? Absolutely, column length, particle size. You guys know how to adjust selectivity? Yeah, but it's always gonna be a guess. I can't guarantee selectivity. They did the hard part. They guaranteed selectivity. So here's a typical example. They're on a 25 centimeter 5 micron, because that's what everyone uses. They're running on a C18 and with acetonitrile water. So I take their method and I make sure I get the same capacity factor as they do. Easy enough, I'll just use the same solvent. I want the same selectivity that they have. Easy enough, I'll use the same stationary phase, mobile phase. I want their same efficiency. That's easy enough, but instead of a 25 centimeter 5 micron, I want to cut the column in half and cut the particle size in half. So if I go to a 15 centimeter 3.5 micron, it's the equivalent efficiency. So I have a method that I hand back to them that has their same capacity, selectivity, efficiency, therefore the same resolution, but the benefit is since my column is 40% shorter, the analysis time is 40% shorter. So without doing anything else, I just cut 40% of their analysis time. And will this work? Absolutely. This is not a could work, should work, might work. No, this is math. If I put the same 3 numbers there, I'm going to get the same number here. And I'll tell you my last trick is everybody runs this column at 1 mil a minute because in 1985 we were afraid to go above 3,000 psi. So with this column, kick it up to 1.5 mils a minute. You now shave another third of the retention time off. So I deliver a method back to them that works in less than half the time of their original method, and that is simple to do. Like I said, companies are happy to pay a lot of money for this because you just cut their analysis time in half. And this works. So another example, 15 centimeter, 5 micron, that was the second most popular column sold out there. Reduce that down to a 3.5 micron, 7.5 centimeter, cut the analysis time by half, cut the solvent consumption in half. In this case, we lose some efficiency because of the dimensions, but remember, efficiency is a square root term. So even if we lose some efficiency, we lose a lot less of the resolution. This does not even take into account UPLC. So if you have a UPLC capability, on this guy, instead of a 15 centimeter column, we go down to a 10 centimeter column, you're at 30,000 plates. You're better than the original method, and now you're 2.5 times faster just based on your column length. So this is an easy, we call it method optimization. It's easy to do, and the trick is keep selectivity the same stationary phase, same brand. Stationary phase, same mobile phase, same pH, same temperature, you'll get the exact same results. And then what I mentioned before is once you get results, crank up the flow rate. Crank it up, and what you learned yesterday is you're going to lose very little in terms of resolution, but you're going to gain a lot in terms of speed. So if you go too far and you lose resolution, no problem, just back it off. So I'm a big fan of cranking up the flow rate. Look at the results and see if it still works. Question? No. Typical flow rates, this is a good reference slide for you. That's neat, but this is the important part of the slide, this equation. So this allows you to translate any column diameter to any column diameter. So if you're running a 4.6 millimeter ID, like the rest of the world, and you decide, hey, I want to try that 2.1 millimeter because I want to save 80% of my solvent, you take a ratio of the diameter squared, and that's going to tell you the factor. So I've done this a bunch of times, 4.6 divided by 2.1, and that ratio comes out to be roughly 5. So that says we are going to cut our flow rate by a factor of 5. So instead of 1 mil a minute on this guy, I'm going to be 0.2 mils a minute on this guy. I am going to cut my injection volume by 80%. So instead of a 10 microliter injection, I'm going to make a 2 microliter injection. Now here's the tricky part. In theory, I need to reduce the extra column volume by 80%, get rid of 80% of the tubing. If you do that, you'll get the identical chromatograph. And by identical, I mean identical. It will overlay the same retention time, same capacity, selectivity, efficiency, same sensitivity, same everything by scaling down. So if we scale everything down appropriately, we will get the exact same results. The trickiest part of that is scaling down the extra column volume. So what I tell people is switch out your green tubing for red tubing, put in the 2.1 millimeter, you're pretty close. You should be up and running. So that's a good one. If you want to do prep scale, if you want to say, hey, I want to collect stuff, let's go up. Going up is the same thing. We go to a 10 millimeter, just take a ratio of the diameter squared, and that'll tell us how to increase the volume and the injection volume and the flow rate. So we could scale that up. I think I mentioned Eli Lilly had 2 meter columns. You don't do method development on 2 meter columns, and you go broke. Even your Eli Lilly, you go broke because you can't run 40 liters a minute all day long. What you do is you do your method development at 1 mil a minute on a little column, and then you just extrapolate up, and you get the exact same chromatogram on any scale. So the cool thing about LC, the column diameter has no impact on the separation. The column diameter only impacts the mass loading, the amount of mass we can put on the column. So we could do the same separation on any diameter column. That's really cool. That means we could go to bigger scales and collect stuff. So match your column length to your desired resolution. If you need a lot of resolution, choose a long column. In this case, 15 centimeter was the longest available for 3.5 micron. We get a beautiful separation of all 7 compounds. But let's say I'm a pharmaceutical company. This is a quality control lab. I'm doing aspirin tablets. I know everything in the aspirin tablet. I always say you sit down with the organic chemist on the very first day. You say, come up with a list of everything that could be present in the sample. So, of course, you have your major compound, your acetylsalicylic acid. Then you say, well, what if it breaks down? Well, then you get acetic acid and salicylic acid. Well, what are the starting materials? You put those in. What are the degradation products? So you're going to come up with a list of like 7 or 8 things. There's not going to be 100 things. It's a limited. It's like, could uranium ever be in my product? Well, no, uranium can never be in my aspirin tablet because it's not one of the byproducts or anything else. So if you say these are the only 7 compounds in the world that I care about that could ever be present, I don't need all that empty space. I could cut the column length in half, cut the analysis time in half, and still have plenty of space. I can keep going. I know it's hard to tell, but this is a factor of 10. We can actually baseline separate all 7 compounds in 0.8 minutes. So by going to a 1.5 centimeter column, by the way, it's this long. It looks like a guard column. And it does work. It's got some plates on it, and it actually does work. So match your desired resolution to the column length. Questions about columns?